Let us discuss the mechanism of the action of restriction endonuclease enzyme. Right? We know that this restriction endonuclease enzyme has a specific sequence which is called as a restriction site or the restriction sequence. So we'll take example of EcoR1 enzyme only. This EcoR1 recognizes a 6 base pair sequence. Recognizes 6 base pair sequence, and the sequence is GAATTC. GAATTC. Right? And EcoR1 cuts always between G and A. Enzyme is EcoR1. Recognition sequence is a 6 base pair sequence, and the sequence is GAATTC. Guanine, adenine, adenine, thymine, thymine, cytosine. Right? This is the recognition sequence where EcoR1 can scan the entire DNA, and wherever it will find this sequence, it will halt it down there and digest or cut the DNA from that point or that region only. Before discussing the details of the mechanism, we need to understand the term that is called as palindromic sequences. Palindromic sequences are those sequences which read same forward as well as reverse, right? So like MOM mom, MOM mom, DAD dad, DAD dad. Such sequences are palindromic sequences only. Restriction sequences or the recognition sequences are also palindromic. Recognition sequences are also palindromic sequences. Let us take this example GAATTC, right? So 5 prime end and this is the 3 prime end. This would be 3 prime and this would be 5 prime. GAATTC. Complementary sequence would be GAATTC. So GAATTC, GAATTC, right? This is again a kind of an example of palindromic sequence only. Now, this EcoR1, let us say we have started an experiment of RE-digestion using EcoR1 enzyme. EcoR1 enzyme recognition sequence is a 6 base pair sequence of GAATTC, and it cuts where? Between G and A. So here it is G and A, it make a cut here. Here also between G and A it will make a cut. Right? Let us see this sequence when it is making a cut between the or within the first strand. G is there, and here we will get AATTC, because the bond between adenine and guanine, that is a phosphodiester bond, it will also break, hydrogen bonds will also break. So G will be separated, 5 prime to 3 prime, 5 prime to 3 prime. Let us talk about the next strand. Between G and A the cut is made. So 3 prime to 5 prime, here only G will be getting CTTAA. CTTAA. This is 5 prime and this is 3 prime. Right? Now, the first cut was made between G and A. The sequence that we obtained is G separated, AATTC also separated, right? And AATTC will come here, AATTC, because there is a cut between G as well as adjacent A. So if you try to read many reference books and try to understand the diagram that they have given is, we can overhang. The overhangs can be produced with the help of such sequences which are called as palindromic sequences, right? So this AATTC will come and attach at this site. That's it. Because CTTAA is intact. So overhangs are produced bonds that are broken down in the overall production of the sticky ends is both. Hydrogen bonds are also broken down, and between this, let us say G and A splitting has been taken place. AATTC different and G different. So phosphodiester bonds also breaks. So phosphodiester bonds also, hydrogen bonds breaks, phosphodiester bonds break, and then both the overhanging structures will be joined together by the action of ligase enzyme. Please refer any reference book or your NCERT textbook for this diagram. Now again, GAATTC, this is 5' to 3'. This is the first mechanism. The second mechanism is GAATTC. Right? Let us say that recognition sequence cuts exactly from the between. Then what we will get? GAATTC, CTT, AAG. Only phosphodiester bonds break. Only phosphodiester bond breaks in case number 2. And the ends that we received are called as blunt ends, because both are of same size. Blunt ends DNA. Right? What will get? GAATTC, CTT, AAG. Sticky ends are generally preferable because it is a kind of overhanging structure. Like, say for example, if this is a structure and like this, interlocking structures are observed within the sticky end kind of thing. While in case of blunt ends, we receive like this. Okay. So easiest one or the most preferable one mechanism is mechanism number 1, where sticky ends are developed. Blunt ends DNA are also used in the recombinant technology, but then the mechanism becomes tedious. There are molecules which are called as linkers and adapters are used, or they are attached to the sequences, and then they are ligated using ligase enzyme. Right? So additional step has been increased. Linkers and adapters stretch of the sequences are also used. So that's why sticky ends DNA are preferred within the recombinant DNA technology. So this is all about the restriction endonuclease enzyme mechanism. Previously we have discussed what is restriction endonuclease, what are the various types of restriction endonuclease, certain facts and figures, the first restriction endonuclease. Now in the next part we will discuss about the separation of the DNA using agarose gel.